Well, today we're going to talk some more about non-Mendelian inheritance. We talked before about codominant and incomplete dominant inheritance situations. Today we're going to look at some patterns of inheritance with even more complex phenotype determination. Just to give you some idea, we'll be talking about skin color, dog coat color, height. We're going to look at two more different ways that inheritance can take place, multiple alleles, and polygenic traits. So let's look at this multiple alleles idea of inheritance. Multiple alleles is what happens in the human blood type, and we're going to get into that a little bit more, but let's just look at what happens when we have a type A mother. Let's say she has type A blood. The father has type B blood. And they have this baby and they test the baby and the baby has type O blood. Is that really their baby? How is that possible? Well, we would expect that if A or B is dominant, the baby should show that trait, right? Be A or B, or if it's codominant, be A, B. I don't know, something like that. That's not what's happening. So what is happening? Before we can answer the question, we need to look a little bit more at blood type. So you guys already know from last section, there are 4 different types of blood for humans, right? We have type A, type B, type AB, and type O. If you'll remember, that represents those different blood types have to do with the different proteins that are on the outside surface, right? Type A blood has this protein or antigen, okay? Type B blood has this. Type AB blood has both on the surface of the cell. And type O blood doesn't have any. So let's look at the possible alleles for the blood type. Now, instead of just 2 different alleles that can be inherited, when we're talking about blood type, there are 3 different alleles. We have this allele. The I represents immunoglobulin. It stands for the word immunoglobulin. It's the immune system related protein. So there are 2 different immunoglobulins. There's immunoglobulin A, immunoglobulin B, and then the little I means there is no immunoglobulin associated with it. These are the 3 different alleles that can be passed on. So let's look a little bit more at these alleles. Immunoglobulin A allele and immunoglobulin B allele are codominant to each other. This allele, the little I, is recessive. It's a recessive allele. It results in no surface proteins. So, if we look at our 4 different blood types again, type A blood can be, if you have type A blood, and that's your phenotype, that's what is expressed, then your genotype could either be one of these. You could either have a genotype where you have 2 immunoglobulin A alleles, IA and IA, or you could have a genotype that is IA with a little I, because remember the little I is recessive. This also will yield type A blood. If you're a person that has type B blood, you could have one of these 2 genotypes, right? Because you could have both IB and IB, or you could have IB with the little I, because it's recessive, and your phenotype would be type B blood. If you are a type AB blood type, and you have both of these different proteins on the surface of the cells, this is your genotype. That's the only genotype that produces type AB blood. And there's also only one genotype that produces type O blood, and that would be this one. You have to have both of the recessive alleles for blood type to be type O. Okay, let's look at our original question. Mother with type A blood, father with type B blood. How do they have this child with type O blood? Well, let's draw a Punnett square for this. Look at the mother on this side of the Punnett square, the father and his 2 allele contributions on this side. What is the mother's genotype? We know that she has to, since she's type A, one of her alleles has to be IA. But what is the other allele? Well, it could either be IA or it could be an I. Either one is going to give her type A blood. Which one is it? Well, since the child is II, because the child is type O blood, right? So the child has to be this. This is the only genotype that will give a phenotype O blood. So that means the mother has to have this as her other allele. And what about the father? The father is type B blood, so one of his alleles will be IB. The other allele could either be IB or it could be an I. Which one is it? Well, once again, if the child is type O and has II, little I, for the genotype, the father's second allele also must be an I. And then we could, of course, fill the rest of this in. IB, I, and this one would be IA, I, and this one would be IB, IA. So we know the mother's genotype. The mother's genotype is immunoglobulin A, and then the little I, the recessive allele. The father's genotype, immunoglobulin B, and the little I for the recessive allele as well. And that's how they got a child that had type O blood with the two recessive alleles. Now, what if this couple had more children? What would their possible blood types be, and in what percentages? You can pause the video if you'd like to think about that for a minute, and then I will answer the question. Okay, if you had a chance to answer that, let's look at the answer. What would this child's, if the child had that genotype, what would the phenotype be? What would their blood type be? Well, their blood type would be AB. And what would this offspring's phenotype be? What would their blood type be? It would be B, right? So we have an AB, we have a B. And what about this one is an A blood type? Because remember, this is the recessive allele, so it's not expressed, just the A. And B and A are co-dominant, so that's why that's an AB blood type. This is A, this is B. And this child we already know is O. So here we are. The results then would be a 25% chance type O, 25% chance type AB, and 25% chance B, 25% chance A. So what about the positive negative trait, like A positive or O negative? What does that positive negative mean? We talked a little bit about that before, but we're going to look at that more closely. The positive or the negative is a second independent gene, completely separate, and it's known as the Rh factor. The reason we call it the Rh factor is it's named after rhesus monkeys. Their name is spelled with an RH, the beginning letters. And the rhesus monkey was the species in which that particular antigen was discovered, and so we just named it Rh factor. We can have Rh positive, that's dominant. Rh positive is the dominant allele. The Rh negative is the recessive allele. Rh positive, the dominant, it means that you have that additional antigen present on your red blood cells. And the Rh negative, the recessive, it means that the antigen is absent. So that's what the naming conventions are. So let's say you're blood type A positive. You have 4 possible genotypes as an A positive. Remember, you have 2 possible genotypes just to be A. You could be IA, or you could be IA little I. Both of those give you the A blood type. But then in addition, we have this Rh factor, this plus Rh factor. You can be homozygous for it, right, and have 2 pluses, or you could be heterozygous because since the Rh positive is the dominant allele, this will also give you a plus and a minus, will still give you Rh positive. So these are the 4 possible genotypes. What if you're blood type O negative? You only have 1 possible genotype. The O is only given by the genotype little I, and since the Rh negative factor is also the recessive trait, the recessive allele, then the only way you can get a negative is by having both alleles be negative because it's recessive. So this is the only possible genotype for blood type O negative. So hopefully that gives you a good idea of this multiple allele inheritance. Blood type is a type of inheritance that is obtained through multiple alleles. We have more than one allele involved in, we have the IA, we have the IB, and we have the little I that are all involved. So we call that multiple alleles. Now what happens, let's look at a different situation here. What happens when a really tall man and a really short lady have children? What do we expect? Well, in Mendel's peas, tall was dominant. But what about human height? Are we just tall or just short like the pea plants? It doesn't really work that way. So let's look and see what's actually going on. Humans can be almost any height imaginable. When we look at, here's just a graph showing U.S. middle-aged male height distribution. It is almost a rainbow. I know it's not exactly a rainbow shape, but that curve, see how that curve goes? We just refer to that as sort of a rainbow idea, right? It's a rainbow of phenotypes that is classic for polygenic traits. That's what we're going to look at now, a different kind of inheritance, polygenic. You can guess probably from the word itself, poly, meaning multiple or more than one, genic, genes, multiple genes. This trait is passed on through multiple genes, more than one. So polygenic traits are determined by the addition, where you have multiple genes that are adding together, usually 3 or more, at least 2, however. And this example, it is not exactly how it works in real life. It is just to give you the general idea of how this might work. So imagine that human height, this was just a hypothetical, is controlled by 3 different genes, A, B, and C. There's our polygenic, right? 3 different genes. Each dominant gene adds, say, 2 inches to the total height. If that were the case, you could say, like, what if our base height was 5 feet 2 inches? So a 5'2" person has all recessive. 5'4" has 1 dominant in the mix, maybe in this combination or in this combination. Whatever combination it is, there's 1 dominant, so they get an extra 2 inches. Maybe 2 dominant here would give them another 2 more inches. Maybe if they had 3 dominant alleles, there are 3 different dominant alleles, they get even more height. And maybe if they're all dominant, they end up being 6'2" because we have all dominant, all 6 of them, adding 2 more inches each time. Again, this is just the general idea that the polygenic traits are additive, and they combine to give us something that is a combination of the different traits. Other examples of polygenic traits besides just height, skin color is one example. Eye color, the amount of pigment that is present in eyes is a polygenic trait. Here are shades of brown hair. If you just look at the different shades of all the brown colors in hair, I'm not talking about pitch black or pure white, but if we just look at all the different browns, it's an additive, it's a polygenic trait. It involves multiple genes to give us the darker or the lighter colors. Animal coat color, and even some of the different traits with animal coats. If you look at different dogs, some of their patterns, speckled patterns, little tuxedo patterns, some of those also are polygenic traits. Now let's take a look at a very special case of polygenic inheritance. It's called epistasis. And you probably know Labrador retrievers come in 3 different coat colors. We have chocolate, yellow, and black. And did you know that a single mother and father combination, okay, they can have a litter, in one litter, all 3 colors of puppies present. So how does this happen? So first let's look at just the black and the chocolate labs here. Aren't they cute, little puppies? Labrador coat color is determined by 2 genes, therefore it's polygenic. We have black labs are going to be either, we'll look at the first gene anyway. We get black labs as 2 dominant alleles, or the heterozygous will give us black. Chocolate labs are homozygous recessive. 2 little B's give us our chocolate labs. Gene 1 determines whether or not the dog makes the big B or little b allele for that pigment, okay? So the first gene is going to determine either homozygous dominant, heterozygous, or homozygous recessive for that brown-black pigment. What about this little guy, though? Where do we get these golden labs? Where do they come from? Well, the little golden lab is due to a second gene, gene number 2. That's why this is going to be a polygenic trait. It's a special case of polygenic. Gene number 2 determines whether or not the dog expresses that brown or black color or not. So this second gene, we call it E for epistasis. Epistasis is when one gene controls the other. So gene number 2 controls gene number 1. We can either have in gene number 2, big E, or we can have the heterozygous, big E, little e. That will give us a black or brown lab, okay, depending on the B genes, okay? So if our second gene looks like this or like this, it's going to let the first gene express itself, okay? But what if my second gene is 2 recessive alleles right here? Little e, then I get a yellow lab. It doesn't even matter what the first gene has. This second gene is going to say that that first gene is not going to be able to express any of its color. So the black and the brown are not expressed. Instead, I get this little golden lab. Advanced ideas, if you want to pause the video, you can just read through this and some really interesting things that you could look into. And I look forward to seeing what you come up with, some research that you have, and we'll see you in class.